I conclude this set of videos with an application of alkene metathesis to biomedical problems. More than 20 years ago, a graduate student in the Grubbs laboratory by the name of Helen Blackwell proposed that the secondary structure of a protein, such as an alpha helix, might be stabilized by stapling together two of the side chains on the amino acids. Helen Blackwell demonstrated her idea by a substitution reaction on the alcohol substituted amino acids, which are serine, with allyl bromide. And then she treated this modified protein with the Grubbs 1 precatalyst to get a good yield of the cyclic alkene. Since alkenes are sometimes metabolically active, she chose a catalytic hydrogenation of the alkene. In this specific case, she used a palladium catalyst. We've discussed the rhodium catalyzed hydrogenation mechanism, but palladium catalyzes hydrogenation by the same mechanism. And what Blackwell, working with Grubbs, found is that by locking the protein into an alpha helical conformation, the rate of its degradation by trypsin-catalyzed peptide cleavage was much slower than the native peptide. Keep in mind that proteins and other biomolecules, which you will study in Chemistry 204, are conformationally flexible. An alpha helix is not always in an alpha helix. The protein is folding and unfolding. And through this dynamic process, when the protein is unfolded, it can react with enzymes that will cleave the amide bonds of the protein. This idea of stable peptides has really taken off, and there was a review article published a few years ago in the Journal of Medicinal Chemistry. Helen Blackwell is now a full professor, independent, at the University of Wisconsin. For this year, I've added one more slide of contemporary interest. Because of the global pandemic from the SARS coronavirus 2, biomedical scientists have been trying many approaches, whether it's vaccine development, testing, or new therapeutic drugs. This group of workers, based at the New York City Blood Center, noted that since SARS-CoV-2 enters the cell first by binding to ACE2 and then entering the cell in that way, they felt that if they could treat the cell with an ACE2 mimic that more effectively binds with the spike protein on the virus, that this will slow the rate of viral entry into the cell, and it allows more time for the immune system to destroy the virus faster than the virus can infect the cell. It's already known that the following residues are essential: glutamine 24, aspartate 30, glutamate 35, aspartate 38, glutamine 42, and tyrosine 41 as being involved with binding to the spike protein. They synthesized a peptide containing 30 amino acids, including these 6 key residues, which are outlined in blue. And you will learn how these proteins are synthesized in Chem 204. They tried several different ways of adding alkene substituted amino acids and found that the following case, New York blood stapled protein 4, gave excellent results with an inhibition constant for entry into a cancer cell expressing ACE2 receptors, a complete inhibition at a higher concentration in a different type of cell, and this stapled peptide also had a very long half-life in human plasma. In the stapled peptide 4, alanine 25 is stapled to phenylalanine 32, and serine 43 is stapled to serine 47. The first staple, separated by 7 amino acids, locks 2 turns of the helix. The second staple, across 4 amino acids, locks 1 turn of the alpha helix. This concludes the videos on alkene metathesis.